All right, so let's assume that this here is the operon. And one thing that is true for all operons is that they have numerous genes being regulated by one promoter and operator sequence. And that's the region of DNA located upstream or before this transcriptional start site here. So in this case, in the lac operon, we have the lac-Z, lac-Y, and lac-A genes all being transcribed. And then we have our operator region, which is abbreviated lac-O, and we have our promoter region. What binds to these fragments, or this piece of DNA? So who are the role players in regulating the lac operon? Lac-I is one of them, so the lac-I binds to this operator sequence here. Who else? Lactose does as well. It has a role in binding to lac-I, and we'll come back to that in a minute. So there's lactose. What else? Cap. Catabolic activating protein. You don't need to know the name. You may also see the name in some books or some resources. We'll refer to this as CRP. What binds to cap? CAMP. Cyclic AMP. What's our last role player? Yeah, the RNA polymerase. So you can take a look at this at a holistic view, but what we're going to start out with is just breaking this down into really just two simple switches, on-off switches. One of these switches is an inhibitor of transcription. The other one is an activator of transcription when bound to the DNA. And so that's what we're going to start out with. Okay? All right. So I'm going to start out by just drawing this again. And let's just talk about lac-I. So lac-I, or the inhibitor here, has a DNA binding portion of the protein. In other words, there are specific amino acids that it has that allow it to make contact with the DNA and put it right before the transcriptional start site. Okay? Lac-I is kind of like a roadblock. When it's present on the DNA here, it prevents RNA polymerase from proceeding with transcription. So this you can think of as our inhibitor of transcription. In what cases is lac-I bound to the DNA? So when it's bound to the DNA, is it bound to lactose or is it unbound to lactose? Unbound, yeah. So in this form here, this would be lactose absent. When lactose is present, what happens is we get a change in the shape of the protein. One in a way that prevents it from binding to the DNA. This is kind of like when going back to something we talked about maybe 5-6 weeks ago now. What is the function of an allosteric inhibitor? Or a non-competitive inhibitor would be the other term we used. Yeah, it changes the shape of the active site. And the lactose molecule here is a lot like that. It's what we would call an allosteric regulator. That's a more general term that we use to describe any of these molecules that change the shape of a protein. So lactose, when bound, changes the shape of this inhibitor so that it can't bind to the DNA anymore. Okay? So it removes that roadblock. What else was I going to say? I guess that's it with that one. Oh, one of the things, this should make some logical sense. And what I mean by that is, if you look at the genes that are being regulated here, again, that lac-Z, lac-Y, and lac-A, these genes are all involved in breaking down lactose into glucose and galactose, two monosaccharides that can then be further broken down to produce ATP. If lactose isn't around, the cell's not going to expend energy to produce these genes. They won't be of any benefit. In order for them to actually have some benefit, the LAC-Z gene, for example, we need to actually have lactose around in the first place. So this is a way to regulate the cell from expending energy or from expressing these genes only in times of need, not when it's not required. Okay? So that's our first role player, and just a simple on-off switch that it's involved in. Our second role player, cap. When does cap bind to the DNA? So what has to be true about its relationship with cyclic AMP? Is it bound? Are the two, when the two bind together, what happens? Yeah, the attachment happens. So the only time that cap binds to the DNA is when cyclic AMP is attached to it. If cyclic AMP isn't around, cap changes shape, becomes inactive. So CAMP or cyclic AMP is another allosteric regulator here. Okay? So it's simply, this system really is just a set of two different on-off switches. And based whether both of them are bound, neither are bound, or one or the other is bound, that can change the expression of these genes within the cell. A couple of other features here about CAP that I want to point out. It has amino acids on it that allow it to bind to RNA polymerase. So this is actually recruiting that RNA polymerase to this site where transcription is going to occur. It's kind of like a molecular beacon, helping RNA polymerase to bind to that site. When is cyclic AMP made, or when is it most abundant in a cell? What's that? It is after ATP synthesis. ATP is our measurement of energy currency, right? And there's kind of this continuum here. So ATP, if it's used as an energy source, if a phosphate comes off of it, that forms ADP. If another phosphate comes off, that forms AMP. And then AMP can become circularized to form cyclic AMP. Oh gosh, great googly moogly. Oh damn. So within a cell, you can assume that there's about a finite amount of all of these four different products. When glucose is present, glucose is kind of what we think of as a big contributor to ATP production. And you can kind of think of this as a continuum in some ways. The amount of ATP produced is inversely correlated to the amount of cyclic AMP that's present. So if ATP levels are high, cyclic AMP levels are low, and vice versa. If ATP levels are low, that it suggests that the cell is in need of additional energy. Cyclic AMP is what we call, or some microbiologists call, an alarm mode. So whereas hormones in the human body are involved in changing gene expression or changing the function of a cell, these alarm modes are essentially small molecules that are around to signal to the cell that they're in need of some sort of gene expression change. In this case, it's an alarm that the energy levels in the cell are low. Okay. All right. So given that, let's go back to this table. What I wanted to do is go back and, in the tables that you guys put together, I ended up looking through them this morning and just adding some things if I felt like there were changes that needed to be made. But I want to go through it now together to just break out and make sure that we're all on the same page. And then we'll talk about this a little bit more. So in which cases here, and instead of looking across rows, we'll look in columns, sorry. In which cases here do we find cyclic AMP being present? Would it be when glucose is present or absent? Yeah, so there's an inverse correlation there. If glucose is absent, we've got cyclic AMP. Yes, yes, no, no. And if cyclic AMP is present, does CAP bind? Yeah. Yes, yes, no, no. When is LAC-I bound? When it's not present, right. So when it's absent. So in those cases where lactose is absent, we'd expect to see LAC-I bound. Then in which cases do we find RNA polymerase present? Typically, yeah. So that would mean, let's see, not here. Not here. Oh. Yes. Yes. Okay. Anybody disagreeing with that at all? Okay. In which case do we find expression then? CAP, right? CAP allows for RNA polymerase to bind. So if CAP is present, we'd expect the RNA polymerase to be bound as well. In what cases do we find gene expression then? So when LAC-I is absent, so that, there's something wrong with this. There might be two things wrong with this. Figure out what's the issue. You may need to walk through all the 4 conditions and maybe even draw them out to model what this looks like. I'm going to give you guys a couple of minutes to figure that out. Okay? I can tell you there is something wrong with one of these answers in the which ones are which conditions is it expressed. There might be something wrong here. Might be. Okay? So start first with looking at the expression. I'll give you 2 minutes. Okay, so CAP has to be bound to the promoter. So are you saying that this is a, I'm going to put this in a different color. You're thinking no, no, yes, no? Okay. Anybody disagree with that? No, we're all good? Based on how we have this written out right now, that's the issue. That is the issue. We'll come back and talk about this. The other thing I want to point out here that you may consider to be an issue, and this is based on, I didn't tell you something. Right here, if lactose is absent, LAC-I is bound to the operator. One of the things that it does is, or one of the ways to think about it is, where LAC-I binds, it actually overlaps with the region where the RNA polymerase binds. And so the two of them are actually in competition for binding to the same region. So for example, LAC-I binds here. If it's bound, it prevents RNA polymerase from actually binding to the promoter as well. So the two of them can't be bound to a single promoter at the same time. And so in this one, you might say here, you might consider this to be a no. Okay. All right. So why is this one a no? We've got that lactose is present, therefore LAC-I would be, or the LAC inhibitor wouldn't be bound. What was the rationale page?
The cap to the promoter.
Yeah, the cap needs to be there to increase the amount of activity. or to recruit that RNA polymerase to the promoter as well. So what we find, just to go back to that model again real quick, what we typically find here is that in order for that RNA polymerase, and this is because this promoter sequence here has a really weak binding site for the sigma factors that recruit the RNA polymerase, but in order for there to be a strong bringing of that RNA polymerase to this promoter, you need cap there to help stabilize it. Okay. Now there are some slight exceptions to this. Sometimes we can observe what we call leaky expression. So if we were to look at this in terms of graphical data, and there are ways to analyze this pretty darn easily. So if we set a graph up of LAC-Z expression or LAC-Z activity on the y-axis, we had our 4 conditions with glucose, without glucose, with lactose or without. What we typically see is the following. If lactose is absent, no expression. It doesn't matter if glucose is there or not. If lactose is present, or I'm sorry, if lactose is absent if lactose is present, glucose is absent, we see lots of expression. This is kind of the optimal or ideal condition. In the case when lactose and glucose are both present, sometimes what we'll see is a little bit of expression, and it's because the RNA polymerase can still bind to the promoter. It just doesn't do so very frequently without that cap protein being present. Okay. And this is that leaky expression then that we sometimes observe here. Go ahead.
So why is cap not expressed as much when glucose is present?
It's not that it's not expressed, but the cap isn't binding to the DNA. Because what does cap require to bind to the DNA?
You said glucose, right?
Not glucose. It requires a lack of glucose. Because what's cap actually binding to? Right, and cyclic AMP is, again, it's a signal that the amount of energy within a cell is low. Okay, so and that's really only going to happen when, in a case where you have 2 carbon sources, glucose and lactose, that's going to happen when the glucose has been exhausted or run out. Okay, so cells typically, and this is true in anywhere from bacteria through yeast at least, we can observe that they will utilize glucose first as a carbon source, and then they will switch to something else, lactose being kind of a secondary one. So what we find is if we're looking at expression over time, you'll see that this lac promoter will be pretty low level of expression, and then all of a sudden peak once glucose has been exhausted and the available ATP has been depleted. So the cell has to find another way to replenish that ATP source. Does that help some? All right, so questions on this process? Okay, let's go to a problem and try this out. We're going to look at this slightly differently now. So I put together a good, better, best question, and the answers are already posted up on Blackboard under today's lesson. But I want to work on this problem that's the big one at the bottom here. How might the regulation of the lac operon change? So how might this affect the activity or the expression of those LAC-Z, LAC-Y, and LAC-A genes if a point mutation occurred that blocked the ability of LAC-I or LAC inhibitor to bind to lactose? Before you answer, I'm going to give everyone a couple of minutes to kind of write out a response or think about this one. One of the ways you can start working on this one, if you need a starting place, start with what you know would normally happen, and then think about how the inability to bind will affect that. All right, go. Sir?
Yeah, what do you
Would it?
Because there's nothing to block it.
Ah, okay. So I see what you're saying. You're thinking of and these do exist. You're thinking of an example where lac-I, the point mutation has occurred, where like it's always in its inactive state, right? And you're absolutely right. That would occur. In this case, what we're saying is the mutation occurs at a location that prevents lactose from binding to the LAC inhibitor. So if that were to take the, so it's actually the exact opposite of what you've said in this case. So in this particular one, what we would find, so if that's our lac-I, it basically has a mutation that prevents lactose from binding. Therefore, what's true about this? What's true about the LAC inhibitor? Where is it going to be found 100% of the time? Always on the DNA here. In that case, then, what happens? No transcription, regardless of whether glucose is present or absent. And so what would happen then is in a system where both glucose and lactose are the only two carbon sources, the cell would utilize glucose, we'd see an initial growth curve, and then it would enter stationary phase, and the lactose would remain present in the media because there's these genes that are involved in breaking it down and using it as a carbon source for energy would never be made in the first place. Okay, does that help to clarify it? Now, in the example that you mentioned, Sid, so there are examples that exist where lac-I has undergone some sort of mutation in the what we would call the DNA binding region. And in this case here, this would always be unbound. So you'd always get expression whether lactose were present or not. In other words, these are examples where the LAC inhibitor is insensitive to the presence or absence of the lactose. Okay, it doesn't undergo that regulation. Questions on that example? Okay, so what I want to do, I want to take about, let's take 3 or 4 minutes right now. And one of the things that I asked you guys to work on with this homework was to actually lay out what are some of the trends that you observe based on the table that you have put together. We've done some modifications to that table, so what I want you to do now is take a look at the trends again. And work with somebody else on this one, and just write down for yourself a few trends. I'll give you a couple of minutes, and then we'll catalog these together as kind of a wrap-up of the conversation on the LAC operon.
Can you just like put the chart back?
Yeah, absolutely. And most of this stuff may be deleted, so I'll have to... Oh, wait, no, it's not.
No, there's one that was.
Yeah, you're absolutely right. Thanks for reminding me of what's on my own sheet.
I got you.
What are some of the major trends that we observe? Good answer. Did we all spend time thinking about nursing interviews again? No. Any trend? Okay, perfect. So polymerase has to be bound in order for expression to even occur. Perfect. That's great. Okay, so must be present for my abbreviation for transcription is TXN, just so you're aware. To occur.
Wait, for what was it?
So RNA polymerase must be present for transcription of these of the LAC-Z, LAC-Y, and LAC-A genes to occur.
So why for this first column it says RNAP combined to the promoter and you put no, but there was still some expression. Is that not?
Yeah, that's a great question, Zach. So the way that I describe it to most people, I showed you guys an example of this leaky promoter that we sometimes see. What I'm going to now tell you is you can think of it that way if you want to. But what I tell most people is if you want to just have this as a binary yes or no type answer, we typically say that no transcription occurs, even in that case when CAP isn't around.
Okay.
Yeah, thanks for asking that. I meant to clarify that. So in other words, the only time where we find expression is when, at least in the context of the lac operon, glucose is absent, lactose is present. That's the only time. Okay. Other trends.
If glucose is present, cap doesn't bind.
Okay, good. So if glucose is present, then no cap binding. Another way to say this would be what? If glucose is absent, cap binds, because cap is in that case bound to cyclic AMP. How about one more? One more general trend from this data.
Is lactose is present, then the lac-I comes on.
Perfect. So if lactose is present, then lac-I can't bind. And there are numerous other ones that you can pick out from this as well. There are numerous other ways that you could state this. But think of this as kind of like our summary of of the major points of today. Okay? Consider this our summary for that. Now, I want to give you one more example. This is not one that I am holding you to knowing, but the operon systems are just one way in which genes can be regulated. And genes can be regulated by promoting or repressing transcription. They can also be regulated after transcription has occurred, so before translation occurs. One example where we see this is actually with some of the porin proteins, or those outer membrane proteins of some of the gram-negative cells. So the mRNA that encodes the OMP-F gene, here's our start codon. Start codon's where the ribosome binds, begins translation, right? Everybody's on board with that still? Nothing changed in our central dogma? There are some RNAs produced by bacterial cells that actually they don't produce any protein. And these are not rRNAs, they are not tRNAs. They're a class of what are called small RNAs. They bind complementary and antiparallel, like so. What will that do to translation? How does this small RNA in red here, and the name of this one is it's called MIC-F. What will the effect of that have on translation of the OMP-F protein? It will. Why? Yeah, it bound to the start codon, exactly. So if the binding to the start codon occurs here, the tRNA can't bring an amino acid to bind to this codon in order to allow for the start of translation. Additionally, their sequences up here, we briefly talked about how with the ribosome, for example, it doesn't only recognize the start codon, it recognizes some of the sequence around the start codon. Well, with that, then, the ribosome can't even bind in this case either. And so this is an example where we see regulation occurring after the gene has been transcribed, but before translation has occurred. And we find this not only in prokaryotes, but we find examples exactly like this in eukaryotes as well. So there's a lot of diversity in how those genes are regulated. Okay, so just to give you a heads up on a couple of things. So for Monday's class, what we're going to cover is mutations, and we're going to talk about it in the context of gyrase again. For the homework, this is another two-parter. The first part's due midnight tomorrow night. Second part's due midnight on Sunday. The first part, this is a blog one, so each member of the group has to pick one mutation that is shown in the blog, and I guess I should pull this up so I can show you exactly what I mean by this.
Is it like in the wiki section?
Yeah, it's actually in a blog section this time, but the instructions for it are right in lesson 4.
I was worried about it all.